still not like the blood vessel that one gets in the body. If you flow blood through it, very quickly it will coagulate, so it's not functional. And that's one of the big problems in trying to make it. So people, us and other people are working on this to try to make more functional-like structures.
Right, but it looks structurally like
It's
right.
getting there. It's early on still.
Right.
So we don't have too many papers on that specific aspect of it yet. Yes?
When you're preparing those gels, do you add some media in it, like for the cells to get feed?
Sure. So the question was about the gels and what solution do we actually put in them. So we actually, so if you want to integrate cells in there, so we put a lot of like we use media as the 95% of the solution in there. But if we don't, if we just want to make gels, then we can put other like water or a saline solution. But when we have cells, we like to fill it initially with media.
Okay.
And so the cells at least initially get the glucose and other kind of signals that they need.
And you keep it, like keep changing it time to time, or it's just
Sure.
one time?
So, and we actually, so let's say we have a piece of gel.
Mm-hmm.
So we put it in a solution of a big bath of media.
Right.
And then we change the big bath of media around the gel.
Okay.
So, but a key issue is that the solution has to diffuse in. And if you have big gels and you don't have too much vascular-like structures in there, then sometimes it takes a long time for, let's say, proteins that the cells need to diffuse in, even a few millimeters. It literally takes a very long time for diffusion to occur. Yes?
Development in nerve tissue?
So the question was about nerve tissue. So we have worked on it a little bit. I think there's a community of people who are interested in using similar kind of technologies for spinal cord repair, where we can try to recreate conduits where nerve tissue can actually start kind of bridging this conduit, and we can put a lot of biological signals in there to kind of induce the nerve cells to do that. So that's there's a lot of interesting work going on there. Some of the technologies which I mentioned can be very easily applied to those things. We've done a lot of work with also making gradients in materials. And gradients are very important for axon growth because the cells actually sense gradients and move along those directions, or even extend axons in those directions. So there's a lot of kind of crosstalk, but my lab is mostly working on some of the other tissues. Yes?
What tissues do you feel like are going to be most successful initially?
Sure. So the tissues that we think are going to be most successful initially are always tissues that are least difficult and least complex. So there's actually very interesting work that has been done in other simple tissues besides skin, such as cartilage, because cartilage doesn't have too much blood vessel supply, so you can actually make pieces of gel with cells in it and then start using them. There's a lot of clinical work that has been going on with bladder tissue, again because it's a biphasic, it's made from two different tissue types, not too much blood vessel supply. So those are, next to skin, those are the next generation of tissues that are becoming more commercially available. The more complex ones like the liver, kidney, and pancreas and heart are much more difficult. And if you can ever do something for the brain, I always say like we're like people who makes like hammers and stuff, and if you want to engineer a brain, you have to be going from hammer to a computer, making from computer. So it's a big step. But people are trying to make neural networks, actually put nerve cells down and then connect them in a very organized way using similar kind of micro scale approaches. So, but I think the complexity, the order will be based on the complexity of the tissue. Any other questions? Super. I know it was crystal clear too. Yeah.
Weeks ago I read the Mattia group and the master converted adult skin cells into blood cells without creating the stem cells and then developing those. That seemed like totally impossible.
Sure.
So what do you think about that?
Sure. So the question was about whether we can take adult cells and then make other adult cells from them without actually having to go through a stem cell intermediate. So that's actually there's a number of papers in those areas that are coming out. One of the first one was from some person in Boston where he took the pancreas cells, the alpha cells, which don't make insulin, and he converted them to the beta cells. So what people are starting to realize is that basically all the cells have the capability to do anything because they have the full genome. They have all the blueprints of doing any other cell type. What one needs to do is to start being able to turn on and off the genes in a very controlled way. So, and what people are starting to see is that there's master regulators that can turn on and off a series of genes based on this master regulator. So what has been the approach is finding these master regulators and then turning them on and off in different adult cell types to just kind of switch the cell to some other fate. And these things are, it's kind of like if you want to see how the cells are, they have particular stable states, and in between these stable states they have a lot of transient states. So what one needs to do is kind of like a chemical reaction. You have to take the cell from this stable state, kind of perturb it to get it to a transient state, and then it will go back to another stable state, which may be different from what it originally started at. So that's the generic biological kind of trans-differentiation approach that people are starting to do. And it looks feasible. So I think it's as feasible as definitely the stem cell approach. All these things definitely need more kind of research to see whether clinically any of them are going to be very useful. Excellent.
Any other questions? Well, if not, let's thank our speaker.